In addition to the anti-stress genes that have been implicated in lifespan extension in Drosophila, there are also some orthologs to genes that we've already talked about in yeast as well as in C. elegans that have been similarly implicated in longevity in Drosophila. So if you remember back to our yeast lecture, you can see here this is the Drosophila ortholog of SIR2, which is that histone deacetylase that can affect gene expression, and an overexpression of SIR2 is able to increase longevity, just like it is in yeast. Similarly, down here, you can see TOR, which is the Drosophila TOR, or DTOR, which is orthologous to mTOR, found in yeast, and that was involved in cell growth, and a loss of function of DTOR, just like a loss of function of mTOR in yeast, can also increase longevity. And then one pathway we talked about in C. elegans is the DAF2 pathway, or that insulin IGF-like signaling pathway. And there are some genes involved in insulin signaling, the InR gene, which is the receptor, D, or Drosophila FOXO, which is the transcription factor orthologous to DAF16 in worms, and chico, which is a IGF or insulin-like receptor substrate, and mutations in these genes can also lead to similar increases in longevity in the way that they did in C. elegans. We're going to talk a little bit more about those insulin signaling genes in the next couple slides. And so the first gene implicated in flies and extension of longevity that's in the insulin pathway is a gene called chico. And chico encodes a chico protein, which is a substrate for the insulin or IGF-like receptor. And chico is actually responsible for a small body phenotype. So loss of function chico mutants are much smaller in size, as you can see here on the left fly, compared to wild type, which is this fly on the right. And that's just a result of having not only smaller cells, but also just less cells in general. And insulin receptor substrates are generally pro-growth. And so you can imagine that a loss of function in one of these substrates would lead to smaller growth or smaller body size. So that's a phenotype we might expect from this loss of function mutation in chico. And additionally, when we look at the survival curves down here for female flies, both wild type in blue and the chico loss of function mutants in red, we can see that a loss of this chico gene, or loss of function of that protein, actually can extend lifespan. And this works through the same insulin signaling pathway that we talked about in C. elegans. It even actually uses the same transcription factor to ultimately affect genes involved in proper reproduction versus entry into a diapause. And in worms, that was DAF16. In flies, that is D-FOXO. Both of these proteins are FOXO transcription factors. So not only does chico loss of function mutation extend lifespan in the same way we saw in worms, it does so via the same mechanism. And in addition to the substrate for the insulin receptor being able to extend lifespan in flies, the mutations in the receptor itself, or the InR gene in flies, have also been shown to increase lifespan. So the InR gene, as I said, encodes an insulin or IGF-like receptor, the InR protein, which is orthologous to DAF2 in C. elegans. And flies that have weak loss of function mutations in the InR gene, or the insulin receptor gene, have not only an increased lifespan, as you can see here, but also reduced egg production. And this has given some researchers kind of make it so that, and so this is something that was also seen, if you recall back to the previous lecture, in flies that are undergoing diapause. So in that diapause state, there's no egg production because the ovaries are not formed. And diapause usually occurs in response to an improper or unfavorable environmental conditions. And so they don't produce eggs during that time, but when environmental conditions improve, they will enter back into the normal life cycle, and their lifespan is extended as a result. And when we look at the loss of function insulin receptor mutant flies, that reduced egg production can actually extend lifespan even further than a fly that went through diapause. But the relationship between reproduction and a decrease in the amount of reproduction and the extension of lifespan is one that's important to point out here. Another thing that's important to point out is that you can actually rescue this loss of function insulin receptor mutation by adding additional insulin receptor substrate, one of which is a growth factor known as juvenile hormone. And so if you actually give flies with a loss of function insulin receptor mutation enough juvenile hormone to supplement any reduced levels in the mutants, they will undergo normal egg production. And once their egg production is back to normal, their life cycle shortens. And so it seems like there's a direct correlation between a reduced egg production in the insulin receptor mutants, and their extended lifespan that can actually be reverted by flooding the system with a bunch of substrate and by reinstituting normal egg production. And so once again, that idea of a trade-off between giving resources to reproduction and the germline towards egg production versus giving resources to the somatic cells of the body is one we have to think about when we think about lifespan, right? Because you can see that both no and reduced egg production will extend lifespan, whereas normal egg production, where a lot of those resources are being devoted to the germline, means they're not being devoted to the soma or the rest of the body, and these flies tend to live or have a shorter lifespan than the others. And this is something that we've seen in yeast as well as in worms, that there's this trade-off between reproduction and longevity.